All right, so hello everyone and good evening to all of you. I am Uroosa Ejaz. I am working as a lecturer in Shaheed Zulfikar Bhutto Institute of Science and Technology, Karachi, Pakistan. And I am also a PhD scholar in the Department of Microbiology, University of Karachi. The topic which I am presenting today in Biofuel 2022 conference is Halophytic plants, an alternative force for biodiesel production. So in this presentation, I'm going to cover the settings, introduction, methodology, research, conclusion, and the team which performed that research. So let's start the introduction. Biofuel is now the demand of everyone because now the petroleum reservoirs are going to be diminished and it is impossible to fulfill our requirement by using the petroleum fuel as a reservoir. So we have to think about the biofuel which can be used in future. There are many types of biofuels: first generation, second generation, and third generation. So how biofuel can be produced? We can utilize the plant biomass. That plant biomass can be converted into sugars by using microorganisms. Microorganisms have the ability to degrade the plant biomass by producing certain enzymes. So when obtained those sugars, these sugars can then be converted into biofuel. So the problem with the biofuel is food versus fuel dilemma, which means that if we use the plant biomass for biofuel production, the food demand will be disturbed. So we have to think about an alternative resource which can be utilized for the production of biofuel. And secondly, the question is, can we produce the enough amount of biofuel by using plant biomass? And what would be its environmental impact? Will it affect our food cycle, food chain, or not? So halophytic plants are the answer of all these three queries. They are basically the plant which can grow in saline condition. So currently, there are no as such usage of halophytic plants. Some halophytic plants although used for medicinal properties, but now the researchers are thinking to utilize that halophytic plant biosource for multiple product formation. And one of the main objectives is biofuel. So halophytic plants are basically the plant which survive in saline environment, and the climate conditions resulted in rich diversity of halophytic plant flora in worldwide. There are around 2,200 species worldwide of halophytic plant, and in Pakistan, around 410 halophyte species are present. So in methodology, we collected the halophytic plant, Phragmites carga, and some other plants. We pre-treated it in order to remove the lignin content, because in plant biomass, the main plant cell wall components are lignin, hemicellulose, and cellulose. So in order to utilize the carbohydrate content, first we have to remove the lignin. So to remove the lignin, two pre-treatment methods were used: acid pre-treatment and freeze pre-treatment. After the pre-treatment of plant biomass, the bacterial enzyme cocktail was used to saccharify the plant biomass. Saccharification is basically the process in which the microbial enzymes act on the plant cell wall component and produce different reducing sugar. So these reducing sugars were then estimated by the DNase method, dinitrosalicylic method. The results showed that among 5 different bacteria, Annually Bacillus, Thermoaerophilus, UE1, DB Bacillus, Porphyrolensis, UE10, Bacillus vesteri, UE25, DB Bacillus, Porphyrolensis, UE27, Bacillus volif mortis, MH1, produce different titers of enzymes. EG and BGL stand for endoglucanase and beta-glucosides, which are basically the component of cellulase enzyme, and xylanase and pectinase. So all these 5 bacteria are capable to produce plant cell wall degrading enzymes in good amount. Especially the Bacillus vesperi UE25 produce the higher titer of enzyme. These enzymes were then used for the saccharification of halophytic plants. Three different halophytic plants were used: Phragmites, Carca, Halopyrum, and GB. Two different pretreatments were performed: acid pretreatment and freeze-thaw pretreatment. So results showed that the maximum saccharification were occurred for Phragmites Carca when it is acid treated, and similarly it is for the Halopyrum and Panicum antidotol. So it means we can conclude that the acid pretreatment is the better way to pretreat the halophytic plant as compared to freeze-thaw method. Because when we pretreat the halophytic plant biomass by acid, it results in the lignin removal. All right, so initially the individual enzymes were used, but then to produce the maximum reducing sugar, the enzyme cocktail were used to sacrifice the plant biomass. Three different bacteria enzymes were used: UE1, UE10, and MH1, and it is used for the three different plant biomass: PK, PA, and GB. So the results showed that the enzyme cocktail comprised of UE1, UE10, and UE25 produce the maximum amount of reducing sugar in case of PA halophytic plant biomass, and in case of Phragmites Carca, the enzyme cocktail of UE1, UE10, and MH1 produce the maximum amount of reducing sugar. The saccharification experiment was performed at two different temperatures, 40 and 60 centigrade, because the enzymes of UE1, UE10, and UE25 are thermostable in nature, and their optimum temperature is 60 centigrade, whereas MH1 optimum temperature is 40 centigrade. After the initial experiments, a statistical tool, central composite design, was used in order to optimize the saccharification condition. Central composite design is basically a statistical tool in which we can select the 5 levels of each factor. So in this study, the temperature, pH, substrate concentration, enzyme units of UE1, enzyme units of UE10, and enzyme units of MH1 were analyzed. 5 different levels of each factor were put into the software. The software provided the 46 experimental run design. The experiment was performed, and the amount of reducing sugar was used as a response. After the analysis of central composite design, the Pareto chart showed that the interaction of factor pH and enzyme units of MH1 has significant effect on saccharification process. Similarly, the interaction of pH and substrate concentration has significant effect. Pareto chart basically that shows us about the significant relation of different factors. These are the contour plots which were formed by using the software. Contour plots showed the interaction of two different factors. For example, here you can see on x-axis we have the pH, and on y-axis we have enzyme unit for MH1. The shape of the contour plot is very important. If it is oval in shape, it means the interaction is significant. But if the shape is not oval, it means the interaction is not significant. So here we can see that the maximum color, dark color, represents the maximum saccharification. So maximum saccharification occurs when the enzyme units gets increased. Similar, when pH gets increased, it has positive effect on saccharification process. So we can conclude that the relation of enzyme units and pH is directly proportional to each other. Similar is the case for substrate concentration and pH. All right, so here you can see the scanning electron microscopy images of untreated Phragmites carca, acid pre-treated Phragmites carca, and sacrificed Phragmites carca. So in native image, you can see the structure is quite rough, but after the pre-treatment, the structure is completely destructed because the lignin content gets removed. And when we perform the saccharification, the pores on the surface can be observed. These pores represent that the enzymes acted on the cellulosic and hemicellulosic content of plant biomass and resulted in production of reducing sugar. And these reducing sugar can be utilized for bioenergy production. These are the graph of FTIR, Fourier transform infrared spectroscopy. Here you can see that when pre-treatment was performed, it results in the removal of lignin peaks. In FTIR, we can analyze the peaks of cellulose, hemicellulose, and lignin. So after the acid pre-treatment, the peaks of the lignin gets removed. And when we perform the saccharification, the change in peaks of hemicellulosic content and cellulosic content were observed, which shows the successful saccharification of plant biomass. So from this study, we can conclude that the enzyme cocktail of comprising the cellulase, pectinase, and celanase enzyme can yield more amount of reducing sugar by utilizing the halophytic plant biomass as compared to single enzyme. Secondly, the acid pre-treatment is more effective as compared to freeze-thawing method. And statistical tool Federal composite design is a valuable tool to optimize the saccharification process in order to obtain the maximum yield of reducing sugar. So halophytic plants growing in a semi-arid region, arid region, saline condition can be a target, can be an alternative resource for bioenergy production. So this is the team which conducted this study, Mr. Ejaz Ansari, Ms. Uroosa Ejaz, and Dr. Mohammad Suhail. Mr. Ejaz Ansari is an MPhil scholar in the Department of Microbiology, University of Karachi, and currently he's pursuing his PhD from China. And here is me, Uroosa Ejaz, lecturer at the SABIS and PhD scholar in University of Karachi. And Dr. Mohammad Suhail is my supervisor. So I want to thank the whole team, and especially to my supervisor, my mentor, Dr. Mohammad Suhail, for his continued support throughout this research work. This is the published paper of this study. The paper was published in Frontiers in Microbiology in 2021. In this paper, you can find out the detail of the experiments. So I want to thank the whole team of the organizers for organizing such an informative conference and for inviting the speaker across the worldwide. So it is a great experience for me to share my study with all of the valuable and scientists and the researchers across the worldwide. I want to thank everyone. Thank you.